Okay, perfect. Now, let's start with chapter 1. First thing you have to know is types of microscope. You have light microscopes and electron microscopes. And from electron microscopes, you have another two branches: scanning electron microscopes and transmission electron microscopes. Now, light can see anything greater than half the wavelength of light. So the resolution, let me also open my notes. 1 second. As I was saying, the resolution of the microscope, which is the ability to distinguish between two separate points, depends on the wavelength. A smaller wavelength means more interaction with anything that's tiny. So a higher resolution, or rather a stronger resolution. Okay. Now, the wavelength of light, or the smallest the wavelength of light can be, is 400 nanometers. So the maximum resolution, the highest quality we can see, is 200 nanometers. So anything greater than 200 nanometers will not be seen by a light microscope. Now, what are these things? Any membrane-bound organelle, except the mitochondria and the chloroplasts, because overall their size is much larger. And the nucleus, obviously, again, because the size is much larger. So, such as the SER, the rough endoplasmic reticulum, Golgi apparatus, and in some cases, some vesicles. Why? Because there's nothing to distinguish between inside and outside. If you cannot see the difference between here and here, you can't, this line is not there. Can you guys tell the difference? No, and generally you won't be able to. And obviously, the ribosomes as well. Their size is 18 to 25 nanometers. Now, for the case of the chloroplast, for example, the inside is much greener because of the chlorophyll pigments. So we know that this isn't the case. We can actually distinguish between inside and outside, so we can see it with a light microscope. Cell surface membranes can't be seen with a light microscope because they are 7 nanometers, and this is a very important number. Okay. Red blood cells can be seen, and so can cell walls. Cell walls are 7 micrometers, so they can be seen. Nuclei can be seen because of the densely packed chromatin inside, so they're much darker, so we can see them with a light microscope. What we can't see with the light, we can see with the electron. Now, the electron microscope, electrons have a much shorter wavelength, as we can see here, so they have higher resolution. Note about the point on magnification. Magnification does not depend on resolution. It simply depends on the microscope, because I can keep stacking lenses on lenses on lenses to get a really high magnification. But if the resolution is not high enough, I'm not going to see anything. It depends. So the ability to see anything, any structure, depends on both the magnification and the resolution, with the stipulation that the magnification does not depend on the resolution or the type of medium, be that as it may, light or electrons. Now, the scanning electron microscope produces a 3D image with more depth, while the transmission, it's a flat 2D image. But for that, the transmission electron microscope has a higher resolution, and that is important. Compare these two pictures. You'll find them in the compendium as well. So we see here, this scanning electron microscope produces a 3D image with depth, with depth, while the 2D transmission electron micrograph only has a 2D image. But for that, the transmission electron micrograph has a higher resolution, and you can also see internal structures because you are slicing, because while preparing the specimen, you slice it in half, almost piercing through the cells. In scanning electron micrograph, you don't slice it as thin. Moving on, magnification questions. If you get a magnification question wrong and it doesn't have like eyepiece graticule or anything, something's wrong and you should probably restudy this part or just solve more. Very simply, of this triangle, actual is image over magnification. Draw the triangle. If you can't do the maths in your head, draw the triangle. It's not a problem. As the magnification increases, the actual size is constant. What happens to the image? Actual equals image over magnified. Image is equal to actual times magnification. The magnification increases and the actual remains constant. What happens to the image? The image increases in size. And now this brings us to the second point, which is the eyepiece graticule and the stage micrometer. Now, we always calibrate the eyepiece graticule based on the stage micrometer. If I have a microscope here, I'm not going to be able to draw it as well as this. If I have a light microscope like so, the eyepiece graticule goes right here. So no matter how much you change the magnification, the eyepiece graticule will remain the same size. It's like a sticker that you just stick on top of the lens. The eyepiece graticule will not change size depending on the magnification. The stage micrometer is here on the stage, and for those of us who have seen it or have used a microscope, as I hope we all have in paper 3, stage micrometer does change size. The more I zoom in, higher the magnification. The actual doesn't change. The greater the image will be. 1 second. Back again. So, say I have an eyepiece graticule as shown, and the stage micrometer as shown. It's just a matter of cross multiplication. Here, let's say each division is 0.1 millimeters. For the sake of example, this isn't constant. 0.1 millimeters is equal to 1, 2, 3, 4, 5, 6 eyepiece graticule, 6 eyepiece graticule units. And you have a cell that's 25 eyepiece graticule units. Just cross multiply to find its actual length. 25 times 0.1 over 6, whatever the answer may be. Now, the key thing here is that the calibration of the eyepiece graticule and of the stage micrometer, the calibration of the eyepiece graticule and its use have to be the same, have to be at the same magnification for this calculation to work out. So same magnification means you just calibrate normally. Now, what if there's a slight difference? Okay, what if, for example, like one question said, one error, like we calibrated on times 10 and use the magnification of times 40. Well, then it's just maths. Now, the image of the eyepiece graticule won't change. As I said, the eyepiece graticule is just a sticker that's like put on top of the, it's just put on top. So no matter how much you zoom in or out, the image does not change. So if actual equals image over magnification, and the image is constant, but the magnification increases, what happens to the actual size? It decreases. So each division of the eyepiece graticule is now less. Less by how much? V times 4. So the calibration will be divided by 4. Alternatively, if you'd like to use the stage micrometer and then adjust the eyepiece graticules on it, 0.1 millimeters, 6 eyepiece graticules times 4. 0.1 millimeters equal 24 eyepiece graticules, or 6 eyepiece graticules is equal to 0.025 millimeters. That's what happens when magnification changes. Now, we'll talk about some cell structures. It's very important for you to know what are the single membrane organelles, what are the double membrane organelles, and which structures or organelles don't have any membranes. Now, the double membrane organelles are 3. Number 1, nucleus. in the name of the organ, in the name of the membrane is the nuclear envelope. Number 2, mitochondria. And number 3, the chloroplasts. Different color because it's a plant cell. What about the single membrane organelles? Everything. Rough endoplasmic reticulum, smooth endoplasmic reticulum, Golgi apparatus, lysosomes, vacuole, vesicles. All of these are the single membrane organelle. All of these, with the exception of vesicles, sort of, are not present in bacterial cells, in prokaryotic cells. Oh, and the membrane of the vacuole is called a tonoplast. Now, what about the non-membrane organelles? It's everything the bacteria has. So, the ribosomes, microtubules, the cell, obviously. That's about it. With ribosomes, we know that there are 2 types, 70S and 80S. When asked, do eukaryotic bacteria by the way, microtubules not present in bacterial cells. Centrioles are also not membrane bound, and they are not present in bacterial cells. That's just a nor are they present in plant cells. So with ribosomes, there are 2 types, 70S and 80S ribosomes. When asked, do eukaryotic cells have 70S ribosomes? The answer is yes, they're present in mitochondria and the chloroplasts. What about 80S? Only eukaryotic. Okay, and these are present in the cytoplasm, free in the cytoplasm, and on the surface of the rough endoplasmic reticulum. Speaking of reticulums, the rough endoplasmic reticulum and the Golgi apparatus are flattened fluid-filled sacs, while the smooth endoplasmic reticulum is tubular. And this distinction is important. You should be able to recognize all these structures from both a light micrograph and an electron micrograph. So I'll send the entire PDF. I'll save you the trouble and I'll just send you the entire PDF. Much easier. Okay, you should be able to recognize all the structures, including, and here's just a quick overview, including the centrioles, which have 9 triplets of microtubules, the flagella or cilia, which have 9 plus 2 structure, 9 microtubule doublets with 2 central microtubules. The complete one is called an A microtubule. The incomplete is called a B. This one has 10 protofilaments, and this one has 13 protofilaments, and this one has 10. And the A microtubule has dynein arms to latch onto the B microtubules of the next doublet, and that's why. And that's the flagellum. Microvilli, they have actin filaments. This is and these are two ways it may be represented. Either from a top view, looking this way, or from a side view, looking left. Okay. And what about plant cells? Here, obviously, we have the cell wall, the nucleus. Again, the membrane of the vacuole is called the tonoplast. You have chloroplasts, plasmidosomata are thin cytoplasmic strands between cells that allow for direct simple diffusion. Now, a few things to note. The difference between cristae and cisternae. I hope you guys know these terms. Do you guys know these terms? Oh, perfect. Okay. Now, in a mitochondria, the inner membrane, 1 minute. The inner membrane is folded like so. Maybe with much better quality than my poor drawing skills. Not just like so. These folds are called cristae. Then what are cisternae? In, whether it be the smooth endoplasmic reticulum, the rough endoplasmic reticulum, or the Golgi apparatus. Forgive my poor drawing skills. Each fluid-filled sac is called a cisternae. And it's important to be able to recognize the difference in an electron micrograph. 1 minute. Sorry for the many brief intermissions. Now, you should be able to recognize the difference in an electron micrograph. Usually, you find that the Golgi apparatus kind of looks like the Wi-Fi symbol. Kind of. The rust in the plasma reticulum is usually either really close to the nucleus, or will have a very clearly defined rough surface. And the smooth endoplasmic reticulum, which is much more irregular, you've noticed, is much smoother, not as rough of a surface, and this will be clear in the electron micrograph when asked to identify it. Prokaryotic cells. All of them have not all, bacterial cells all have a peptidic glycan cell wall. Circular DNA, plasmids, 70 S ribosomes. They are obviously unicellular, and they are 1 to 5 micrometers in diameter. Does this mean they can be seen by a light microscope? That's a question. Murine is peptidic glycan. It's the same thing, it's just different names. And obviously cytoplasm. All cells will have a cytoplasm and the cell surface membrane. These are the minimum requirements for a cell. To have cell surface membrane, to have a boundary and something inside it. I should probably speed up. That's, oops. Circular DNA and 70 S ribosomes and plasmids. And they are 1 to 5 micrometers in diameter, so they can be seen by a light microscope, and you should be able to compare between prokaryotic and eukaryotic cells. I'll do viruses with infectious diseases. Biological molecules. I'm not going to talk about the tests. You've memorized these in paper 3. I'm not talking about them. Carbohydrates and lipids. Okay, let's start with alpha glucose. Sorry. All right, sorry, let's start with beta glucose, and I'll tell you why in a minute. Beta glucose, first thing we note is the OH is up, and then we just alternate. OH, H, and then we go the opposite. OH, sorry. OH, H, OH, H, OH, H, and finally CH2OH, H. Alpha glucose is exactly the same, except for one tiny change. We'll switch these. Now, because it's an alpha glucose, nothing's going to go on top of it. It's alpha. If it's alpha, it's the first. There's nothing that's going to be above it. There's no OH above it. Okay? So if you ever are asked to identify the alpha glucose structure, look for alternating OHs, except for the first. For the second, they're all just alternating. Up, down, and this is up. You don't. It's paper one. You're not going to draw. Okay. In paper two, you can be asked to draw the glucose. Okay. Now, what about monomer? Now, monomer is the unit that forms a polymer. What's a monosaccharide? Monosaccharides are there are three types of monosaccharides: trioses, which are intermediates; pentoses, which are two types: ribose and deoxyribose, and hexoses, which are three types, and you should know this: glucose, fructose, and galactose. The structural formula is CnH2nOn. So trioses, C3H6O3. Pentoses, ribose would be C5H10O5. Deoxyribose means minus one oxygen. Deoxy minus one oxygen. So C5H10O4. Slight change. Hexoses are all C6H12O6. Now, what about the disaccharides? You only have two in your syllabus. Maltose, which is glucose plus glucose, and sucrose, which is glucose plus fructose. Now, what is the structure? What is the formula for these two? Very simply, C6H10O6 plus another one, C6H12, sorry, minus H2O, because when they're joined, it's a condensation reaction. Sorry, it's a hydro. Yeah, when they're joined, it's a condensation. Water is produced. So that's just going to be C12H22O10. 6 plus 6 is 12. 12 plus 12 is 24. Minus 2 would be 22. 6 plus 6 is 12. Minus 1. Sorry, that's 11. Would be 11. Okay, and what about polysaccharides? Now, in polysaccharides, you have three types. Glucose, you have four. Amylose, which is a single helical chain. Amylopectin, which is a bunch of amyloses together. So is this helical? No, it's not, because normal helices don't have little branches coming off them. No, it's not. Nothing I'm saying is out of syllabus, or unless you're talking about something else. Mesosomes are out of syllabus. Now, myelopectin, it's not a straight chain, it's not helical. Okay, but it is more useful because I have more free sites for energy. And finally, glycogen, which is just amylopectin taken to the max. It is really branching. Then you have cellulose, which is just a straight chain. Now let's come to cellulose and have a little discussion. You have the molecule, the microfibril, and the fiber. Not to be confused with collagen, which we'll discuss later. Now the molecule, simply alternating, connected by beta 1,6 glycosidic bonds. Obviously, cellulose is beta glucose, and the other three are alpha glucose. Beta 1,6 glycosidic bonds joining glucose molecules, so they have a zigzag shape or just a straight chain. The microfibrils. Now, because of this, they have OHs everywhere. I know I'm not drawing. I know I'm not drawing a proper type, but you get the point. Like a proper diagram, you get the point though. These form hydrogen bonds with the ones below them and above them. So when multiple cellulose molecules come together, they form hydrogen bonds, form microfibril, and then multiple microfibrils join together in parallel to form a cellulose fiber. Now, cellulose fibers then have a crisscross shape in the cell wall to make a crisscross shape to make the cell wall, and these gaps allow water to pass through it. That's right, beta 1,4. And here, this has alpha 1,4, and these have alpha 1,4 and alpha 1,6. Yeah, it is beta 1,4. Sorry, my mistake. Now, what about triglycerides? They are hydrophobic, non-polar molecules. So they have glycerol, CH2OH, CH3OH, CH2OH, CH3OH, and fatty acid chains. The fatty acid chains are simply hydrocarbon chains with a carboxyl group at the end. They condense together. They condense together to form the triglyceride. Now, because the triglyceride has many C-bond H bonds, that means it's very good at storing energy. Okay, it's also an insulator, so it can be used for thermal and electrical insulation. That be in neurons or in to protect sensitive or for thermal insulation and to protect sensitive organs. What about phospholipids? And we'll discuss this in the next chapter. Actually, you know what? I'm just going to discuss it in the next chapter. Now, proteins. Amino acids have a general structure that you should know. Central carbon. Carboxyl group off to one side. Amine group off to the other. A hydrogen here and an R group here. And this R group will determine the property and the name of the amino acid. Now you have the different types of structures. Here. Okay. To make a protein, the amino acids join together by a what happens? Normal condensation. Water is formed, or the liquid condenses, goes as to liquid. So liquid is formed, usually water. So condensation reaction, water is formed. Hydrolysis means I need water to lyse a substance. Lyse means break down. This forms a peptide bond, and the bond itself includes 4 elements: C double bond O and N bond H. So if it asks you whether for the secondary structure the bonds are the atoms involved are part of the peptide bond, yes, because we have this double bond O and this bond H, and this we saw in March 26. What about the structure? Its primary structure is the sequence of amino acids. It's the first thing made by the mRNA. And the secondary structure, alpha helix and beta pleated sheets. You can read these in your own time. Tertiary structures, 3D shapes that are functional. Yes, it will be uploaded. Okay, and you tertiary structure, all proteins reach at least the tertiary structure to be functional, with an active site here and a specific 3D shape. And they are held by interactions between R groups. So primary, polypeptide bonds. Secondary, beta pleated secondary structure. Hydrogen bonds between C double bond O and H bonds and N bond H, more specifically the O and the H. Whether these amino acids are adjacent, i.e. opposite each other, or they are 3 to 4 amino acids apart. Tertiary structure, interactions between R groups, one polypeptide. Quaternary, more than one polypeptide chain. They can be identical though. So I could have exactly the same sequence, copy paste, and they're just bonded to each other. That's possible. Okay, here we have a small example of which structures are seen versus which structures are not seen. These structures will be present regardless. They go in sequence. So if you have quaternary structure, but you must have a tertiary and a secondary structure. But in this example, I see single chain of amino acids, so that's primary structure, and I see 2 different polypeptides held together by R group interactions, that's quaternary structure. I don't see secondary and tertiary, but in your life, they'll always be there. If you have tertiary, you're going to have secondary, so you're going to have primary, regardless. Now, these are the types of interactions. We already talked about the peptide. Hydrogen bonds are present in all structures. It's a weak bond. Ionic bonds, they make the tertiary and the quaternary structures, and they are between C double bond. They're between CO minus, which are polar charged R groups, and they are broken down by pH change, decreasing the pH, increase the number of H plus. H plus is going to bond with C double bond with COO minus. Increasing the pH, so increasing OH minus, it's going to bond and H3 plus. Then you have disulfide, which is between. Say, what about methionine? I know it has sulfur too. It does not have the proper thiol group, which is bond which is SH. Whatever. Okay, so methionine does not form a disulfide bridge. Only cysteine does. And then hydrophobic interaction, simply nonpolar R groups, they push the water between them. The weak bonds, the only strong bonds are the covalent bonds. So disulfide and peptide. These are the strong bonds. Hydrophobic, really, really weak. Hydrogen, weak. Ionic, sort of weak. Now, globular versus fibrous proteins doesn't really come a lot. Concerning collagen. Collagen, there are no microfibrils. You have the molecule. You have a polypeptide. Every third amino acid is glycine. Now, does this mean that all 3 polypeptides have to be identical? Not necessarily. They can be different. Okay, the important thing is every third amino acid is glycine. Why glycine? Because it's the shortest one. This R group is just 1 hydrogen atom. Where were we? Okay. Everything glycine, which is the smallest amino acid, for tight coiling. These now they could be different. The other amino acids, they don't have to be glycine. They could be anything else. Okay. All three polypeptides together make up a triple helix, which is held by hydrogen bonds. Then you have triple helices, and this is the molecule. The collagen fiber is these molecules held by covalent bond. Okay, and they have the staggered end. Why? Here is the collagen. If they don't have a staggered end, they're lined up nice and all I have to do is just break this, and the entire collagen fiber gets unraveled. With the with a staggered end, we are avoiding weak points. Why? I break here, nothing else gets broken. I break here, nothing else gets broken. I break here, nothing else gets broken. So the general structure of the molecule remains bound. This is the same. Here is a more specific diagram. This is the molecule, and this is the fiber. Here are all the together. Moving on to water. Water is dipolar, so it acts as a solvent. Usually in these questions, they're going to play a trick on you. They're going to give all the correct information, but they're going to ask you for specific use and say which of these, we saw this in Feb March 26, which of these is useful. If I am simply transporting a substance from one area to another, do I need there to be high latency to vaporization or high speed? No, you don't. But I do need it to be dipolar, so it acts as a solvent. You should know that there is a maximum of 3 that there is a maximum of now there is a maximum of 4 hydrogen bonds that a water molecule can make with another water molecule. Okay, but each water molecule, so one water molecule with another water molecule, that's just one bond. Okay, one hydrogen bond. There is none of this stuff. Okay, this doesn't happen. Obviously, we just talked about it being bipolar, high specific definition. Now, cohesion and adhesion, very important. Cohesion is water molecule, hydrogen bonding with another water molecule. Adhesion is water molecule, hydrogen bonding to the water molecules, hydrogen bonding, sorry, to the hydrophilic regions of cell wall. Hydrophilic parts of the cellulose cell wall, the cohesion, water molecules together. This is important. A small part of lignin is hydrophilic. Most of it is hydrophilic. I'm jumping to connect ideas. Onto enzymes. They're globular proteins, and that means they are unchanged. Biologicals made in living organisms, catalysts to speed up chemical reactions without being used up or chemically changed, and they can be reused. There's a lock and key. There are 2 mechanisms: lock and key mechanism and induced fit. Lock and key, fully complementary, binds by temporary hydrogen bonds, forming enzyme substrate complex, but it's no longer fit, they are released. Induced fit, not complementary, they enter, change, if there's a conformational change of the enzyme, so it molds around substrate, so stronger and better fit and bind. Everything else is on a reading basis. Let's just move to the inhibitors really quickly. Competitive inhibitor, same Vmax. Why? The maximum rate of reaction remains the same. If all the enzymes manage by some coincidence to latch onto a substrate, that's the maximum that's going to happen. At high enough substrate concentration, substrate wins. Okay, so there's the same maximum. However, at lower substrate concentrations, there's a different rate of reactions, so there's a lower affinity for the enzyme to the substrate. Simply what happens in induced bit is the substrate comes close to the active site. It makes interactions with the R groups. Now the R groups change, and this causes the enzyme to change shape. So it molds around substrate, so there's a stronger fit and bind. For more effect, so a better enzyme or a more efficient enzyme substrate. What about non-competitive inhibitors? Non-competitive inhibitors, they have the same effect. The enzyme work are not somehow divided between one substrate and another. No, they're going to work at the same exact. They're going to work. They're going to have this. However, some enzymes are permanently ruined, while the non-competitive inhibitor is present. So the Vmax, the maximum, it's never going to happen that all the enzymes will have a functional active site. So the maximum possible rate will not be achieved. That is the difference between competitive and non-competitive inhibitors. On to immobilization. Immobilization makes it easier to purify, more thermostable, and more stable to changes in pH, and easier to extract the product. And they can be reused. I put them into a little container like this, so I don't have to fetch the liquid enzyme or the aqueous enzyme from the milk or whatever. Okay, these are the advantages. Obviously, it's cheaper because you get to reuse them. Self-signaling doesn't really come a lot. You just read it or watch one of the previous sessions. DNA. Here we get to the interesting part. We're in chapter 5. And we have, okay, hopefully we'll manage to speed up. Did I skip something? I skipped cell membranes and transport. Oh, yay. Okay, really quickly. This is really important. Someone asked me to ask them, but I forgot who it was. So can you guys just like figure it out? Important thing. When talking about diffusion, time taken to fill an agar, how many? Question, show of hands. When to use surface area to volume versus when to use smallest diffusion distance. Perfect. Okay. So here I have two. I'm going to give you a 2D representation, but 3D. Now, since these are made of the same material, time taken for diffusion to move a set distance does not change. Okay, so in both materials, it takes this amount to move this amount. In both, all that needs to happen, is to go from here to here, across the entire thing. The signs don't really matter, because and this is why we use diffusion distance when asked for the blocks to be completely filled. Okay, the same thing happens here. It's going to take the exact same amount of time, and this answers one of the questions that was sent, to reach the center, to completely fill the cube. Is there something coming from the sides? Yes, but because by the time I reach the exact center, this would have managed to drop, and this would have moved the exact same distance. Here, by the time I reach the center, it's going to be somewhere over here. It doesn't matter because I'm already done. Does it make sense now? The minimum distance is the maximum distance that the substance needs to move. Now, as for when do we use so if I have a block like this, and if I have another block like this, okay, even though this probably has a surface area to volume ratio that's higher, I'm not going to do the math, but probably there are certain cases where this is true. All that needs to happen is this moves this tiny distance, okay, across the entire thing. It just moves a tiny distance and it gets to the center and the block is complete from both sides. They just meet in the middle, okay, the entire block is complete. Not true. You don't use the surface area to volume ratio and you see, no. Here it has to move a much larger distance, so it's going to take more time. We use surface area to volume ratio when we want to compare the efficiency of diffusion. Let me see if I can, 1 second. Okay, so if I have this block and I have, oops, no, this block. If I stop them at a specific point in time before they're completely filled, I'll see that this has a higher percentage filled than this one. Now they're going to be close because this is a 2D representation, but in 3D, it is going to matter. Now here, it's 8/9 of the thing is filled. What about here? We have 3 by 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. So that's 30, 1, 2, 3, 4, 8, 22/30 of it is filled. Now, which one is filled more? Obviously this one, if you go ahead and calculate the percentages. Exactly, thank you for the question. So here, it's asking for the complete diffusion. So we're going to use the minimum distance of 5 by 10 by 15, the smallest is 2.5 millimeters from both sides. Same thing as the first block that we have, 5 by 5 by 5, the minimum is 5. Okay. However, if I stop both blocks at a specific point in time before they are completely filled and ask which one is filled, that's when I use the surface area to volume ratio. Here it's 8/9, here it's 22/30. However, when asked when they'll be completely filled for the complete diffusion of the dye filling the block, then I use the minimum distance, and usually minimum distance is the one you see with these types of questions. If you calculate the surface area to volume ratio of 5 by 5 by 5 and 5 by 10 by 15, you'll note that 5 by 5 by 5 has a much higher surface area to volume ratio. So if we're going by surface area to volume ratio, 5 by 10 by 15 will require more time. However, they both have the same minimum distance. So by the time they both reach it, it's okay. I use surface area to volume ratio when I'm looking for efficiency. Here, in the same amount of time, most of this block's covered. Here, much less percentage. If you're going to go ahead and calculate it, do so on a calculator, find that I'm right. This is the concept. Here, in a set amount of time, I move 1 block. Here, the same amount of time, I'm going to move 1 block. Okay, from both sides. Both sides. Here, a higher percentage is covered than this. But by the time this reaches 1.5 blocks here, from all 4 sides, it's going to take the same amount of time. This doesn't have to cross. This can just reach 1.5 blocks like so, but this will cross 1.5 blocks from all sides. Same thing here. Okay, and it'll be filled. So this side doesn't really matter. Like this length doesn't really matter. Do you guys understand this? Okay, moving on to fluidity of cell surface membranes. Cholesterol has a really rigid structure. Really rigid structure. I didn't understand. Um, Moving on. Can you guys hear me now? Perfect. Okay, I'm going to describe these questions again. Sorry for light mode one more time. Here it asks for the surface area to volume ratio, so it's not even a question. Here, where the idea is that through the same substance, at the same amount of time, the substance will diffuse the same distance, provided that the diffused substance and the medium through which it diffuses are the same. So, however, the percentage cover is different for C. And the answer here, I assume, is C. If you get out a ruler, you're going to find the distance from the edge to the colored part in both the 1 centimeter cubed and 3 centimeter cubed and the 2 centimeter cubed is the same. However, we realize that because the 1 centimeter cubed has a higher surface area to volume ratio, a higher percentage of it is empty or colorless. While for the 3 centimeters cubed, since it has a lower surface area to volume ratio, more of it it still has the color. So there is less efficient diffusion with 3 centimeters cubed and more efficient diffusion with 1 centimeter cubed. Here I explain this question since it asks for the complete diffusion of the dye. We're working by minimum distance. 5 by 10 by 15. Minimum distance here is 5. We're going to look at the block with another minimum distance of 5. The answer is the first block, which is 6.2 seconds. So the answer is A, 6.2. Is that okay? Oh, no screen. Okay. Can you guys see the screen now? Okay, perfect. Again to the questions. The first question, it asks for surface area to volume ratio. You're just going to calculate it normally. This isn't, the block that becomes colorless in the least amount of time, smallest diffusion distance, obviously 2 centimeter by 2 centimeter by 2 centimeter. It's not even a question. And it's going to have the highest surface area to volume ratio. Like this is a really basic question. Okay. For the second question, again, we're working by the premise that for the same substance, through the same diffusion medium, it diffuses the same distance in the same amount of time. So I'm going to go and get my ruler and measure this distance right here from the edge to the colored section, 2 centimeters cubed, and replicate it across the 1 centimeter cubed and 3 centimeter cubed. So I'll find that my answer is C. Why is that? Because same amount of time, same diffusion distance. Okay, and we take the minimum distance. However, since 1 centimeter cubed has a larger surface area to volume ratio, it has more efficient diffusion, so a higher percentage of it has the HCl already diffused, and so is colorless or whatever the color is. While 3 centimeters cubed, lower surface area to volume ratio, less efficient diffusion, so it takes much more time so it takes much more time for the entire cube to be filled. But again, the distance from the boundary to the color is the same across both. The difference here is the efficiency of diffusion relying on the surface area to volume ratio. Regardless here, it's not really that needed. Complete diffusion of dye, immediately I default to minimum distance. Okay, 5 by 10 by 15, minimum distance is 5, so I look at the same for the block with the 5 centimeters cubed. The answer is 6.2, so the answer here is A. I hope that was good. Anything else? Okay, perfect. Back to the session. Semi-conservative DNA replication, you're just going to memorize it as is. Don't forget the idea of anti-parallel strands, and don't forget the idea that DNA polymerase works continuously from 5' to 3'. Well, here what happens is it does, let's just say it starts here. It does 5' to 3', and this area is just unzipped. So again, we cover the next area. And then the following area is unzipped. So the DNA polymerase goes all the way back and covers the next area, and that's what forms the Okazaki fragments. Okay. Since the DNA polymerase requires 2 strands to work, this is just some extra information. We add a teeny tiny bit of RNA primer here that it latches onto and continues. DNA polymerase must start with holding two different strands. Why the telomeres shorten? Okay, talking about telomeres and telomerase. Which cells have telomerase? Cells that divide rapidly and continuously. So, stem cells. Of course. What else? Cancer cells. What else? Activated B lymphocytes. The anti-lymphocytes activated. I'm just going to write activated lymphocytes. And why is that? Because they're going to undergo clonal expansion. So by histones are going to replicate. So by cytokines, they're going to replicate really rapidly. By stimulation due to cytokines, they're going to replicate rapidly. So we're going to need telomerase to ensure that DNA is not damaged and these cells don't become a problem. What other cells? Nothing. Yeah, activated B and T lymphocytes. I don't think there is anything else. Talking about, okay, now this is really important for the mitotic cell cycle. I'm sure the notes are here somewhere. Okay. You should have this memorized. Gap 1, or the first stage, G1, you have the normal cell functions: exocytosis, endocytosis, protein synthesis, which I'll cover in a few, and the buildup of ATP to support all these. Then semi-conservative DNA replication, S phase, makes sense. And then in G2, we are in G2. Okay, 1 second. I want to change something. We're going to take this and put it here. Okay. So G2 is preparation for mitosis. So the mitochondria and the chloroplasts divide frequently. There is a huge buildup of ATP for mitosis, and centrioles divide. We said we're going to move that to S phase. Then you have mitosis and cytokinesis. Mitosis is nuclear division, not cellular division. So bacteria do not perform mitosis; they perform binary fission, because mitosis is a nuclear division. Bacteria don't have a nucleus, so how can they have a nucleus divide when they don't have a nucleus? Makes sense. Mitosis is for growth and asexual reproduction, tissue repair, cellular replacement, and the immune responses we discussed for clonal expansion. Small notes here, do not confuse these two. The mitosis does not repair cells. It replaces these cells. These cells have a problem. Okay, we kill them. Bye bye. We get other new cells to fix. So we repair tissues by replacing damaged cells. Growth and asexual reproduction, obviously. Now, prophase, metaphase, okay. Now I want to talk to you about chromosomes, chromatids, chromatin, telomeres, and chromosome arms, because they're going to ask this a lot. They have asked this a lot. Chromosome is simply the strand, okay? A chromosome is simply either, it may have one chromosome, it may have one chromatid or two chromatids, but it is simply the strand of DNA, okay? Upon S phase, that strand replicates to form two chromatids. Okay. My internet's somehow improving. Yeah, okay. Here. Chromatin is the uncoiled form, uncoiled form. It's the form of the chromosomes during late telophase and interphase. Okay, that's chromatin. Chromatin is the genetic material that is uncondensed. Chromosome simply chromosome is simply the one continuous strand of DNA. Not one continuous strand, one molecule of DNA, sort of. One set of molecules of DNA. So this is a chromosome, but so is this. Okay? Chromatid is one molecule of DNA. When it's condensed, we call it a chromosome. When it's uncondensed, chromosome or chromatin, both work. But condensed, exclusively chromosome. Now, a chromatid is one continuous DNA molecule. Okay, your entire genome is not just one DNA molecule that's very large. No. Each chromatid is its own little DNA molecule, like so. So that's one chromatid. That's another chromatid. That's another chromatid. During semi-conservative DNA replication, okay, the chromosome gains another DNA molecule. So now each chromosome has two chromatids. Okay. Now, what about telomeres? Telomeres are simply caps. They are found here, 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 and here. They are found at the edges of chromosomes. All the edges of the arms of the chromosomes. So if it is just so, it's going to have 4 telomeres. If it is if it has only one chromatid, it's going to have 2 telomeres, and so on and so forth. This is the basic idea of what's the difference between a chromosome, a chromatid, and a chromatin. Okay. Basic guideline. Okay. Chromosome is the specific sequence of genes. Okay. One to 2 DNA molecules. Okay. Chromosome is a specific sequence of genes with 1 to 2 DNA molecules. It has 1 centromere for the most part. Chromatid is 1 DNA molecule. Okay, and the chromatin is simply uncondensed chromosomes. Can you guys still hear me? Each chromosome can have 1 or 2 chromatids, but generally chromatids are visible only during mitosis, because that's when they condense. And please note, it's going to play a lot with the word visible. So you're going to have to note that. Telomeres, they prevent the loss of genes, very simply. Okay. Let's move on to protein synthesis. Very important and very repeated question. You have 1 chromatid. Okay, anaphase is a bit strange because people debate whether it is technically a chromosome, or because people debate whether the separated chromatids are 1 chromosome or 2. So I'm just going to Generally, the number of chromosomes does not change. So let's keep it that way and say that the DNA molecule has 2 separated chromatids. Okay? No, Again, two separated chromatids for one chromosome. The number of chromosomes does not change even in mitosis. Okay, it's a bit weird, I know. Number of chromosomes does not change. What changes is the mass of genetic material, and this depends on before or after a space, and if it says the mass of genetic material in the nucleus, then now you're going to have to pay attention to prophase, where it becomes 0 because there is no nucleus, and in telophase, in which it divides by 2, and so on and so forth. These are the things you're going to have to pay attention to in the questions. They like to play with this topic a lot. Protein synthesis. The pathway of protein synthesis is incredibly important, and I'm sure all of you have seen at least one question that talks to you about we added radioactive amino acids. What happens to from what pathway, or what lights up first, or what becomes radioactive first? First things first, the amino acids are going to be endocytosed or diffuse into the cell, regardless. So the first thing you might see it in is vesicles. Endocytic vesicles, not endocytic vesicles. Yes, DNA mass does not change in telophase. I'm saying in a nucleus, the DNA mass per nucleus might change. But overall, no, the DNA mass does not change except in S phase and cytokinesis, obviously. Now, the pathway for protein synthesis is important. First thing that happens, endocytosis. The amino acids are taken in by endocytosis. Great, perfect, moving on. They go where? To the ribosomes to be assembled. Then rough endoplasmic reticulum, then vesicles again, then Golgi apparatus, finally secretory vesicles or lysosomes. Secretory vesicles go out of cell, lysosomes go into cell. You have many pathways here, okay? So, and they're going to play with it a lot. Endocytic vesicles first, then ribosomes, whether that be for the rough endoplasmic, or whether those be on the rough endoplasmic reticulum or the membrane cytoplasm. Then rough endoplasmic reticulum, then Golgi apparatus, then rough endoplasmic reticulum, then packaging vesicles, then Golgi apparatus, then secretory vesicles, then out of cell bye-bye to the radioactive ore, then secretory vesicles or lysosomes. Okay, semi-conservative DNA replication. I want to talk about what did I want to talk about with the yes, evidence. Vesicles are moved through by the cytoskeleton. The cytoskeleton is kind of like roads that the motor proteins use to grab the vesicles and move them. Okay, they don't move through the cytoskeleton, they move by it. You don't move through a road, you move on the road. I did not, no, not with emphasis. I described the basic concept, but I did not go any further. Semi-conservative DNA replication evidence. Okay, very important, focus here. We have 2 DNA strands, and normally 2 DNA strands, okay? Then we introduce radioactive isotope, whatever it may be, N15, whatever the question says. We're going to move through it. Now, the first thing that happens depends if the amino acid starts in the cell, or if they tell you we introduced it to the outside and then we introduced it to the, and then it was endocytosed into the cell. So I'm going to do this. Put endocytic vesicles in brackets. There you go, better? Okay. Now, what happens is the radioactive isotopes are used in the formation of the free activated nucleotides we talk about in semi-conservative DNA replication. I'm going to make them light blue. So upon S phase occurring, this strand becomes radioactive and this strand becomes radioactive. So for each DNA molecule, 50% of it is radioactive. Okay, moving on. And in this stage, none of this, and starting here, none of the molecules, okay, are 100% non-radioactive. All of them have at least one radioactive strand. Moving again, each strand, this strand, then you have this strand from here, and again this strand, and again this strand. Again, DNA replication. Bum, 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 bum. We went from 50% radioactive, or 50%, we went from, sorry, 50% normal. Here it was 100% normal. To now 25% normal. And note this, you can draw, okay? This cell, DNA molecule and this DNA molecule both have one normal strand. So if they ask you, what is the percentage of DNA molecules with normal strands without specifying a specific percentage, then you say, in this case, 25% strands. Here, it's 100%. Here, it's 100%. And this goes on, okay? So then you're gonna have this, 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 oops. And now the percentage of DNA, total DNA that's not radioactive would be 12.5%. The percentage of DNA molecules with any strands that are non-radioactive is now, oh, sorry, here it's 50% of the strands, two of the molecules, 50% of the molecules have strands that are non-radioactive. Yes, exactly, that's the question I'm talking about. The nitrogen 1415, the radioactive isotope. Here, 12.5% of the total DNA is non-radioactive. How many molecules have strands that are non-radioactive? This 2 strands out of 2/8, that's 1/4, that's 25% of the molecules have strands that are not radioactive, and so on and so forth. This is the basis of how this idea works. Okay, now that's what I wanted to discuss about in semi-conservative DNA replication because it comes a lot. One of the things I forgot to mention in chapter 1, sorry about that, is the endosymbiotic theory. Oftentimes it may ask you about spinning cell organelles, and I'm going back to chapter 1 really quickly, and then we're going to continue with chapter 7, 8. 8's going to have a lot of discussion, and we'll discuss 9 just a slight bit later. Okay, question. We're back to chapter 1. As I said, yes, we do need to know the nitrogen 1415 experiment. I just explained it. You can watch the recording. Moving back again, I'm going to go back to chapter 1 for a small little tidbit I forgot, the endosymbiotic theory. Often you may see this topic called cell fractionation. Okay, in cell fractionation, I don't know if you guys have seen these questions or not, but they are there. What happens is we get a cell, we break it down to its separate, or yeah, exactly. We have a centrifuge, we break it down into its separate organelles, and we spin it really, really quickly, and then it asks what's up and what's down. Exactly. Okay. The idea is the heavier organelles are going to move down, the mass of the pellets, sure. The heavier organelles will move down. The lighter organelles will move up. What is the lightest organelle we have? Ribosomes, exactly. So they're going to be at the top. What is the largest and heaviest organelle with a lot of dense genetic material? The nucleus, exactly. The nucleus. We're not going to separate them into nucleolus and chromatin and whatever. Okay. What about what's in between? Okay. Bacteria, mitochondria, and chloroplasts, all three have exactly the same mitochondria and chloroplasts all have mitochondria, chloroplasts, and bacteria have exactly the same mass. Why? Because mitochondria and chloroplasts were once bacteria, and they were endocytosed into a cell, taken up by a cell. What is evidence for this? Number one, circular DNA. We know that eukaryotic cells do not have circular DNA. However, bacteria do, and mitochondria and chloroplasts do. So now eukaryotic cells have circular DNA within these organelles. 70S ribosomes, same thing. Oh, by the way, when it says hybrid DNA means half and half. Like one strand is one strand is yeah, one strand is normal and one strand is radioactive. Normal, radioactive, normal, radioactive. It's the same thing as this one, as these numbers, exempting the first one. Okay, here, for example, let's do this question. I'm going to open. Okay. Bacteria are grown in a medium containing nitrogen. After several generations, all of the bacterial DNA contained 15N. So it's the opposite. Instead of introducing it to radioactive, we replaced. We had it all radioactive and then introduced it back to normal supply. Same concept. Some of these bacteria are transferred to a medium containing normal isotope, and they were allowed to divide once. The DNA of some of these bacteria was extracted and analyzed. All the DNA was all hybrid DNA containing amounts of 14N and 15N. Okay, so this is the first stage. 100% hybrid. We divide it one more time. How many molecules are hybrid? Again, this is hybrid and this is hybrid. It has one normal strand and one radioactive strand. These two are radioactive, or sorry, these two are normal strands, depending on the question, radioactive strands, whatever. So 50% of the molecules are hybrid. So the answer is B. Chloroplasts do weigh heavier than mitochondria slightly, but overall they're about at the same level. Okay. Obviously, then before the chloroplasts and the mitochondria have all the rough endoplasmic reticulum, rough endoplasmic reticulum first, then the Golgi apparatus, and then the smooth endoplasmic reticulum, because the rough endoplasmic reticulum also has ribosomes on it. So rough endoplasmic reticulum, Golgi, smooth endoplasmic reticulum, and so on and so forth. That's the overall. Okay, you still don't understand. Again, let me open the question. Let me just save. There we are. Here we are. Okay. So first off, medium containing the radioactive isotope, all the DNA was radioactive. 100% of the DNA was radioactive. Then we transferred them to a non-radioactive isotope. Okay, so they're surrounded by a medium that is non-radioactive. Okay, and this medium is the one that's going to be used to make the nucleotides in semi-conservative DNA replication. These bacteria were allowed to divide once. This is the first division. The DNA was extracted and analyzed, and this DNA was hybrid. Hybrid DNA meaning it has one strand that's not radioactive and one strand that is radioactive. So 100% of this DNA is hybrid. Then let's move on. We left it and allowed it to divide one more time. So one more division to 4 molecules. How many DNA molecules here are hybrid, meaning they have one normal and one radioactive, or one radioactive and one normal? The answer is 50%. This molecule, remember, each of these is one molecule. There are 2 strands. Okay, this molecule and this molecule out of all 4 molecules, so that's 2 over 4 molecules, are hybrid, meaning one strand with N15 radioactive and one strand with N14. However, some questions may ask, how much of the total DNA is radioactive? In that case, it's 25%, because only 2 strands out of a total of 8 strands are radioactive. But here it's asking for hybrid molecules. This is a hybrid molecule, and this is a hybrid molecule out of a total of 4 molecules. So 50% of the molecules are hybrid. This is the basic concept. Make sense? It doesn't matter, it's just the same thing. This is the first generation. Okay, first generation, second generation, third generation. You're just going to repeat it one more time. Yeah, there's no way I'm going to draw that. Okay, here we are. I caved anyway. I'm going to do it next, okay? So it's clear. Okay, here we are. These 2 strands are hybrid, okay? So we have 12.5 hybrid molecules. But how many strands are radioactive? 1 strand, 2 strands out of a total. 1 minute. Hello? What's the amylose is hmm. Hello? Can you guys hear me? No, I'm breathing. Okay, perfect. Um, what were we saying last time? I forgot. Someone remind me. We were at the end of chapter. Okay, chapter 7 really has no surprises. I mean, chapter 7 is not at all surprising. Chapter 8 is important, and I want to cover something really quickly. For those who studied my notes, by the way. Sorry. Anyway, I'm going to stop pleasing myself. Hydrogen bonds are between, no, hydrogen bonds are between collagen polypeptides. Covalent cross links are between, no, opposite. Here. Oh, great, my internet's acting up again. Chapter 7 really has nothing surprising. Moving on to chapter 8. Key thing about chapter 8 is the idea of the oxygen dissociation curve. Note this. High partial pressures, high saturation, more firmly bound oxygen, so I get more oxygen to transport. But at low partial pressures, I want a really low saturation, so I can easily release this oxygen. That's just the main idea. This is just the idea of the conformational change. The first oxygen molecule binds. This is difficult. The whole hemoglobin molecule is distorted and many bonds are broken. This makes the second and third easier. The fourth is just slightly harder. Okay, because hemoglobin is always saturated. So it goes to the first part, finds an oxygen, goes to the second, finds another oxygen, goes to the third, finds a third oxygen, finally finds the last place for the oxygen. That's the reason for the sigmoid curve. Not really that common in paper two, but who knows? Okay. Here it's just the concept, like how the structure is overall. Key thing to note is the chloride shift since hemoglobinic since HCO3 minus goes out, a chloride shift moves into the cells for electrical neutrality to make sure that inside and outside the cell are balanced in terms of neutral bond, hydrogen bonds between amine, carboxyl, covalent between the species. Again, hydrogen bonds form the quaternary structure of collagen. Covalent bonds form the cross links between collagen molecules. I'm sorry for jumping around because of the recordings. For the recording. Okay, so what happens is aerobic respiration produces carbon dioxide. 5% of the carbon dioxide goes to plasma, combines with water, hydrogen carbonate ions remain. Okay, 85% of it, which is most, okay, combined with water inside the red blood cell to convert to be converted to carbonic acid by carbonic anhydrase, which dissociates into H plus and HCO3 minus, and the HCO3 minus will still go outside the cell. And the last 10% are just going to bind to hemoglobin and terminal amine groups to form carbaminohemoglobin. And this term is very important. It's carbaminohemoglobin, not carboxyhemoglobin. Okay. Carbon dioxide on the terminal amine groups. Let's see this question. Okay. Which set of labels is correct? Again, if you have the picture in your head, you're going to memorize it. Carbon dioxide in plasma moves into the red blood cell by diffusion from high concentration to low concentration. Obviously not facilitated diffusion. Okay, so A is wrong because carbon dioxide is non-polar. It's hydrophobic, so it can easily diffuse through the phospholipid bilayer, through the hydrophobic core, so 1 is wrong. Okay, B, 2 is water, correct, forming 4, which is carbonic acid, whatever. 8 is dissociation. Oxyhemoglobin dissociates to form oxygen, maybe. 10 is carbaminohemoglobin, no. 11 is carbaminohemoglobin, not 10. 3 is carbonic anhydrase, correct, because it catalyzes the reaction. And, well, okay, yeah, let me label them, much easier. Okay, 1 is simple diffusion. 2 is water. 3 is the enzyme carbonic anhydrase. 4 is the product carbonic acid. 5 is dissociation or breakdown, forming 6, which combines with something, and 7, which exits. That means that 6 is H+, which combines with hemoglobin, and 7 is HCO3-. Okay. 9 produces oxygen. Okay. So must be oxyhemoglobin. 6, and this releases or makes hemoglobinic acid. Okay, 9 combines with 6. The product of 9 combines with 6 to make hemoglobinic acid. Now this involves adding on this carbon dioxide would form carbaminohemoglobin. And it is the process of dissociation or release or whatever you want to name for it. Okay. 1 facilitated diffusion, no, simple. 2 is water, yes. 8 is dissociation, yes. 10 is carbaminohemoglobin, no. And how do I know this? Because carbon dioxide is added to 11, not to 10. 3 is carbonic anhydrase, yes. 9 is oxyhemoglobin, yes. 11 is carbaminohemoglobin, yes. The answer is therefore D. 5 is carbonic anhydrase, obviously not. 5 is dissociation. 6 is hydrogen ions, yes. 12 is a red blood cell, obviously. I didn't even see the label, but yeah. And this is also simple diffusion, by the way. It's the same one here and there, by the way. So, things to keep in mind. You don't get 9 and 11. Okay, I'm going to erase this section. It's very confusing because of the way they laid it out, but here, let me do this. 9 would produce 4O2 and something. 6 produces something. When combined, they produce something else. And we're going to add CO2 like so. and something else. Now, this something else, we're going to have two copies of it. We're going to add CO2 to one of them. Okay? Now, this is 10. You know, let me just take this out again. 9 is going to produce something else. 6 is something, it's going to combine with that something else. Okay, to form 10. And we're going to have two copies of 10. 10 here and 10 here. And we're going to add CO2 to form 11. This is this part. I think this way it's much clearer. 9 produces oxygen. 9 gives out oxygen. What in the red blood cell gives out oxygen? There are no mitochondria. What did I just say? There are no, there's nothing else apart from oxyhemoglobin. Okay. Now this will release oxygen in form normal empty hemoglobin. And we know that 6 is H plus from dissociation. H plus plus HB, H plus in hemoglobin would form hemoglobinic acid. Okay, so we have here hemoglobinic acid. Hemoglobinic acid. We're going to add to it carbon dioxide. That's going to form carbaminohemoglobin. How about that? Is it clear now? The diagram here is very confusing. Let me just erase this section. Again, 9 releases oxygen and combines with 6 to form another product. Okay, 6 is H plus, and we know 9, if it releases oxygen, that's oxyhemoglobin. Oxyhemoglobin, remove the oxygen. You're left with hemoglobin. Plus H plus, that's HHB. Okay, and this we're going to multiply it by 2 to get another hemoglobin, and we're going to add carbon dioxide to form exactly, that's number 11, because it says here, carbon dioxide in plasma moves into 11. Perfect. These are the confusing parts of chapter 10. Yes, this very important at the alveolus. Many. Okay. I was gonna cover this. Okay, let's just do it now. Here is your alveolus with its squamous epithelial cells. Sorry for my poor drawing skills, you're going to have to survive them. And here is the capillary endothelium. And here we have a simple red blood cell. For an oxygen molecule, okay, again, one more time. The hemoglobin diagram, one more time. Do you understand 9? You don't, 8 is just the process. Okay, do you understand 9? What in the red blood cell releases oxygen? Oxyhemoglobin. Exactly. So 9 is oxyhemoglobin. That's clear, right? I know that this, like the way they laid it out, it's a bit confusing, but 9 is oxyhemoglobin. Great, so that's 9 done. We know that 6 is H plus, right? 9 and 6 combine together to give us 10. What's 10? 9 is oxyhemoglobin. We already removed the oxygen from it. So it's like we have 9 that gives us two products, oxygen and something. Okay, we're going to take that something and combine it with 6 to give us another something. And then we're going to take that same something and add to it carbon dioxide. To give us a fourth something. This is how it looks like. Okay. 9 releases oxygen, so it's oxyhemoglobin. We remove the oxygen. It becomes just hemoglobin. 6 we know is H plus. H plus hemoglobin would be hemoglobinic acid, HHB. We add to it carbon dioxide to give us carbaminohemoglobin. That's 11 and that's 10. Clear now? Exactly. To the terminal part of hemoglobinic acid, it doesn't matter. The details don't matter. Here we have carbon dioxide binding to 11. Carbon dioxide binding to this mess, which we know involves hemoglobin. Obviously, it's going to be carbaminohemoglobin. Carbon dioxide, you don't even have to like conceptualize it. Okay. Here, in this section, carbon dioxide is bind to 6 comes from the dissociation of carbonic acid. Exactly. Okay. Carbaminohemoglobin. 1 second. Sorry about that. Okay. No, carbaminohemoglobin binds to a hemoglobinic acid. Let's just fix that. Not necessarily, but plausibly. More commonly. Carbaminohemoglobin binds to HHB. Let's keep it at that, okay? I think that makes everything clearer. That is chapter 10. The cardiac cycle, really quickly. We know that the sinoatrial node first sends waves of excitation that pass through to the atria. AVN delays it by a microsecond, and throughout the AVN, there's a band of contractile fibers. From the AVN, we get the bundle of His, which then branches off to the Purkinje fibers or Purkinje tissue. It's important to note that blood flows like so. Makes a nice V shape and gets out then of the thing. Here, this graph, very important. Okay. First things first, atrial systole. Okay. Pressure in the atria increases and in the ventricles. Then the AV valve closes then pressure very slowly in the atria decreases enough. So in the ventricles, it shoots upward. Ventricles are contracting. This is ventricular systole. Then ventricles are relaxing, pressure is decreasing. That's ventricular diastole. Semilunar valve closes, and there's the recoil of elastic fibers, so that's the little increase. And the atria are slowly filling up with blood till it's greater than ventricles. So there's ventricle increase. That's the hello? Oh, okay. That's the basis of it. Blood vessels, really, there's nothing you should worry about. Just this part, and you should know the composition. Don't forget, in elastic arteries, you will clearly see elastic tissue as like black zigzags in the tunica media. Otherwise, it's probably going to be muscular. You will see very clear, very thick. Yes, we have immunity and diseases. Immunity, I'm not going to cover extensively because there isn't much creativity there. Ah, okay. The graph colors. Sorry, I should probably add a key. Okay. Black is atrium, blue is ventricles, and red is artery, or aorta in this case, and the left side of it. Okay. Immunity, I'm just going to ask you guys what's so confusing. Same thing with infectious diseases. The problem with immunity and infectious diseases is they require more on your critical thinking than on your understanding of the topic. For diseases, I'm just going to emphasize, memorize these better than your name. Please. These are really, really important, and they come up. Monoclonal antibodies don't come a lot in paper one, but sure. The respiratory system, we covered everything there is to cover, just the advantages of this, and I'll send the one more time. I'll send it on the ASAL channel. Actually, for the structure, that's basically what you need to know. And you should know that the elastic fibers here stretch on inhalation. Now, infectious disease and immunity, I want you guys to understand, I want you guys to ask me exactly what your problem is, because it's a very specific thing. Monoclonal antibodies, really, all you have to know is is their formation, and then they act just like any other antibodies. The key thing is, DOS is direct observational treatment. Short term, it's how we treat tuberculosis without having to trap a person in a hospital. You have a person, okay, B lymphocytes and T lymphocytes. You have a person, TB is very contagious, so rather than keeping them trapped in a hospital, some worker or some responsible family member goes every while to make sure the person's taking their meds, because it's very long and it's very tiring and most people just give up. So rather than trapping them in a hospital to make sure they take their meds, you just have someone like visit their house to make sure, much better. B lymphocytes and T lymphocytes. B lymphocytes, B format. Now the name tells us the site of maturation. B lymphocytes mature in the bone marrow. T lymphocytes mature in the thymus. Okay. What happens is, for both, the antibody receptors on them, the antibody receptors on them at the site of at the site of maturation are selected. If it is complementary to a self-antigen, to one of my own antigens, then no, I kill that cell. Because in the end, it's just random. The antibodies are random. That's why some people may have better immunity to some diseases than others. It's just random. Okay, so if by chance it manages to hit one of my own self-antigens, I kill it. If it doesn't, if it hits a non-self antigen, then I keep it alive. T lymphocytes, there are two types, T helper and T killer. The CD receptor is what helps it bind to cells, because T helper and T killer bind to cells, not to not to the antigens directly. Okay. concerning the humoral and cell-mediated immune response. No. CO2 binds to hemoglobinic acid. Carbon dioxide plus hemoglobinic acid forms carbaminohemoglobin. All right. T lymphocytes. Okay. In the end, by the end of their maturation, okay, B lymphocytes they go to secondary lymphoid organs and to the blood. So the spleen, the lungs, lymph nodes. Sorry about that. The T lymphocytes, the lymphocytes in general, they go to lymphoid organs and then to the blood to spread around and to do their function. Okay, now let's just go over the questions that you guys put in the channel. Difficult and spelling MCQs. Okay, let's start from the top. Here, which row is correct for mitosis in plant cells? Plant cells do not have centrioles. Very important. Plant cells do not have centrioles. In prophase, centromeres are present, of course. Centromeres. All linear chromosomal DNA has centromeres. Each chromosome is visible as two chromatids, also correct in prophase. After S phase. Spindle fibers formed by centrioles. Again, no centrioles in plant cells. So A and D are immediately eliminated. I'm not even going to read the rest of the options. For metaphase, chromosomes align at the equator. Yes. Centromeres attached to spindle fibers in metaphase? No. They attach to spindle fibers in prophase, and they are fully arranged at the equator by metaphase. So C is wrong. In anaphase, okay, no. Centromeres attached to, okay. That's a bit strange. Centromeres are attached to spindle fibers. It is C, which is why I'm confused. Centromeres are begin attaching in metaphase. They are attached. Okay, sure. Centromeres attached to spindle fibers throughout metaphase. That's true. Then chromatids separate and migrate to opposite, and chromosomes decondense in telophase. Correct. Centromeres are arranged at the equator by spindle, and they are fully arranged by the end of metaphase, which is why the term centromeres attached to spindle fibers is a bit weird since they are already attached, which is why I'm a bit confused. But C is the most right because chromosomes do not replicate to form chromatids and there are no centrioles, as we said, in plant cells. So A, B, and D are wrong. We work by process of elimination in certain cases. B lymphocyte and T lymphocyte both are initially produced in the bone marrow, and then B lymphocytes remains in the bone marrow while T lymphocytes goes to the thymus, and then they both end up in blood in the lymph tissue and secondary lymphoid organs. Second question, what is happening at time X? At time X, we notice that the pressure of the ventricle is increasing above the aorta. So pressure of the ventricles above the aorta, semilunar valve opens to allow this pressure to end up somewhere, to allow the blood to end up somewhere. So the semilunar valves open, and obviously the atria are during this time, while the ventricles are contracting. Are the atria contracting, or are they still silently filling up with blood? They are filling up with blood. Okay, so the atrium is filling, it's not emptying. Where would it empty? We know that the atrioventricular valve is closed. because the ventricles are contracting. Okay. So the atria cannot possibly be emptying because there's nowhere to empty to. Meanwhile, the ventricular pressure above pressure, so the ventricles. So ventricle contract means atria filling, and atrioventricular valve closing means atria are filling, they're empty. But there is no such thing as the ventricles closing. AV valve close, atrium filling. Ventricles contracting is atria filling. Okay, very simple question. You should just have the graph memorized and the pressure of the ventricle is above the pressure of the aorta. This means that the semilunar valves are forced open. And at this time, obviously, the atrioventricular valves are closed. So the blood in the atria cannot empty because there's nowhere to empty to. So instead, they're filling. Moving on here, this question is really important. I'm just going to take it because there's going to be a lot of maths. Let's just keep it like so, like this. There's no need. The gene that codes for a protein is 130 kilobase pairs long. That's 130,000 base pairs. Every 3 base pairs are 1 amino acid. We know this. Protein R is 220 kilodaltons in mass. The typical mass is 110,000. So you have 220,000 daltons in mass over the typical mass of an amino acid, which is 110, gives us 2,000 amino acids. Which row is correct? Now, what we're searching for is the length of the introns and the length of the mRNA. The length of the mRNA is the mature RNA. I cannot emphasize this enough. When it says mRNA, we're talking about mature mRNA. The mRNA without the introns. So we know each amino acid, 3 base pairs. So we multiply this by 6. That gives us 6,000 bases. Multiply this by 3 based on this rule. That gives us 6,000 base pairs. So the answer is either A or C. What about the length of introns? We know that there are 130,000 base pairs in the gene that contains introns. We remove the introns from them and we're left with 6. That's 130,000 minus 6,000 equals the introns, which would be 124,000. So our answer is C. This question, pretty interesting too. This is some information about the polypeptides that make up hemoglobin. What can be concluded about the information about cysteine in hemoglobin? More than 1% of the amino acid cysteine. Let's figure that out. The position of cysteine in the polypeptide chain of alpha globin, there is one position, so there is one cysteine in alpha globin. There are 2 positions, so 2 cysteines in beta globin. So here, there are 6 cysteines total. Why? Because there are 2 alpha globins and 2 beta globins. So that's 2 and 4. 6 total cysteines. Out of how many amino acids? That's 2 times 141 plus 2 times 146. Let's calculate it. 6 over 2 times 141 plus 2 times 146, which is the total number of amino acids. That would be all times 100 to get the percentage. 1.045. So more than 1% is cysteine? Yes, barely. More than 1% is cysteine. In alpha globin, there's a cysteine close. So A is correct. So the answer is A. Okay, more is more. Doesn't depend on how much. B, C, and D, we're going to read them and eliminate them. Alpha globin, there's a cysteine closer to the end with an unreacted carboxyl group than beta globin. What do we mean? How can a carboxyl group be unreacted if it's not at the end of the polypeptide? All carboxyl groups will react to form the peptide bonds. So B is immediately wrong, unless it's at the end or at the start of the peptide. And neither of these are at the end, which would be the 141st amino acid or the 146th amino acid. So this is wrong. The replacement of cysteine at position 93 decreases the affinity of hemoglobin for oxygen at low pH. If replacing cysteine with a different amino reduces the Bohr shift, meaning higher oxygen, how is this question telling us that it would decrease the affinity for oxygen? And this is obviously wrong. Region of beta containing oxygen 93 more things that faces out when no oxygen is inward when we know that globular inward is facing inward. So when oxygen it faces inwards, so it's less hydrophilic, hydrophobic. Am I still lagging? Okay. Perfect. Again. I don't know how to fix that. The screen is blurred. I don't know what to do. Cysteine is an amino acid. You didn't get the high. Okay. In globular proteins such as hemoglobin, hydrophilic regions face outward and hydrophobic regions face inward. This is for all globular proteins. Hydrophilic out, hydrophobic in. So here it says that this region faces outwards, so it's hydrophilic when no oxygen is present. It faces inwards when oxygen is attached, so it becomes hydrophobic when oxygen is attached. The binding of oxygen to the heme group causes this region to be more hydrophilic. This is wrong. It becomes hydrophobic. It doesn't become hydrophilic. I hope that explains it. Moving on to this question. The region, we're not talking about the entire protein, we're talking about the specific region. It's soluble, sure. I did not deny that. But the specific region that we're talking about, okay, faces inwards. Oh. 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 Can you guys just like I don't know if okay, I'll forward the question that I'm solving. I think that'll be better, so you guys could just follow along. Okay? Sure. Better? So you guys just know what question screen problem. Moving on. Some molecules fluoresce when illuminated. That means they shine. Changes in the structure can change the intensity of fluorescence. The enzyme tryptophan synthase fluoresces bluish green because of the phosphate with the active site. So the enzyme and its active site has a phosphate group, and this causes blue-green fluorescence. Adding serine to the enzyme increases the intensity to the fluorescence, but when indole is added to the bluish-green fluorescence, decreases in intensity. What may be concluded from these observations? Serine and indole attach to the active site of the enzyme. One second. Sorry. Okay. Yes. Perfect. Okay. So, first off, both of those affect the phosphate in the active site. So both of them attach to the active site. It makes common sense. An enzyme serine complex is formed. Correct. Correct. Why? Because serine attaches to the active site. So serine increases the intensity when it binds. So an enzyme serine complex is formed. Here, but when indole is also added, that means we have serine, and then we're going to add to it another indole. So an enzyme serine indole complex is all three statements is correct. The answer is A. Doesn't matter what type of inhibitor it is. It's just that it's not non-competitive because it binds at the active site. The question's here. So if my internet's lagging, you guys can just like follow along with my voice. Two, because adding serine only increases the intensity. When indole is also added, so I add serine and I add indole, so I'm adding two things. Serine and indole. Non-competitive does not bind. Indole is, it's not competitive and it's not non-competitive. It's a third thing. It's called uncompetitive. Don't worry about it. Indole is an inhibitor because it decreases that. It doesn't matter if indole is an inhibitor or not. It changes the phosphate group. It changes the shape of the active site. It doesn't change the shape. It hides the phosphate group. It does something to something at the active site. Okay, both attach to the active site of the enzyme. An enzyme serine complex is formed? Yes. An enzyme serine indole complex is formed? Yes. The answer is A. The answer is 1, 2, and 3. Again, both affect something in the active site, so both bind to the active site. We know that serine binds first based on the question. Adding serine increases the intensity. But when indole is also added, so serine plus indole, so enzyme serine indole active site, so enzyme serine indole complex formed. Moving on to the next question. The diagram shows stage of a tulip's growth. Which row identifies the correct? Or the tulip bulb is acting as a source only or as a sink? The bulb is this region. Okay? It's acting as a source only when there is nothing to provide sucrose. So in what stage? In one, it's not really acting as anything. I'm going to be a bit more specific. One doesn't really act as anything because there is no, it's just it's just a leaf. And the leaves will act as a source. So one is also eliminated. I'm sorry about that. I mixed up. There is no place for it to act as a source. Can you guys still hear me? Okay. So in one, where is it going? There's nothing up and nothing down. So there's no place for so there's no place for the energy to go. There's no place for the sucrose to go. So one is eliminated. And we know D is eliminated because of 5, and B is eliminated because of 4. It acts as source only, so the only answer is C. Notice, in 1, where's the sink? If it acts as a source, there has to be a sink. Where's the sink? In 1. There is no sink. Right? 2, there's some roots, so there is a sink. 3, there is a sink. 4, there are 2 sinks. 5, there stopped being a sink. Okay, now the bulb itself is the sink for storage. 6, the bulb itself is the sink. 7, there is no sink. There are roots here. There are the beginnings of roots in 2. 5, it isn't. 5, it's a sink. I made a mistake. Because it is the sink. The bulb itself is the sink. Why is 5 no? Why is it not a source? Because this is the source, the leaves. The bulb is just there for storage. Okay? So the answer here is C. Again, 1, I'll redo this question from the beginning because I got confused. 1, there's nothing above and nothing below. There is there is no sink for the source. No. 1 is where are the roots in the shoot? Where is it going? Where is the energy going? Can you see roots and shoots in 1? I can't. So 1, no. Leaves cannot. The sentence that roots can be a source when leaves can't produce energy is wrong. Okay. In 2, there are roots. Okay, so there's a sink. In 3, there are roots. There's a sink. So source. In 4, there are roots and the leaves that is not above, or a shoot that is not yet above the soil. So it cannot yet photosynthesize. So there are 2 sinks. And the bulb is still the source. Here, we have a lot above. So here, this acts as the sink. Again, it acts as the sink here because there's a leaf. There are leaves. Yes, the leaf looks almost dead, but okay. 7, it's just whatever. Okay, 7, it's preparing for a new cycle, so it's a source, but not really explicit here. So it's a sink only in 5 and 6. We know 1 is eliminated. We know D is eliminated because of 5. And we know B is eliminated because 4. So the answer is C. Moving on to the next question. Okay. This one. What is the minimum number of phospholipid layers? It didn't say bilayer, so we're not assuming that it's a bilayer. Okay. So here is the drawing. Every cell membrane is 2 layers. Why? Because it's a phospholipid bilayer. Okay, so assuming that molecule crosses cellular layers by passing through cells, so it's going to pass through each cell, what is the minimum number of phospholipid layers that it must pass through? Okay, so it's going from air into alveolus to hemoglobin and red blood cell. So first things first, we have the alveolar epithelium, the capillary endothelium, and the red blood cell membrane. It enters the alveolar epithelium, that's 2. It exits it, that's another 2. It enters the endothelium, that's 2. It exits it, that's another 2. It enters the red blood cell, that's the final 2. 2 plus 2 plus 2 plus 2 plus 2 is 10. The answer is D. Here, last question. Because it's a phospholipid layer, not bilayer. The question says phospholipid layer. So since each cell surface membrane is a bilayer, it has 2 layers. It crosses 10 cell surface membranes
So it crosses 5 cell surface membranes. Yep. Honest mistake. Sorry. Crosses 5 cell surface membranes. And so it enters one cell. So it enters the cell and exits. Enters the cell, exits it, and then enters the red blood. 5 cell surface membranes. 5 times 2 layers per membrane is 10. Last question. Someone deleted it. Either that or I'm blind. Question was deleted. That's it. We're done. I hope you guys enjoyed the session. It was very long. Good luck. Don't stress on your exam. And I know all of you are going to do. Okay, send it. Send the question. I'm going to pause real quick. You're going to send the question. Yes, you can. A 40 is achievable, and paper 1 can help you improve your grade if you didn't do like. I know I'm going to get like absolutely buried when I say this, but it is possible to get a 40 out of 40 in paper 1. I'm sure of it. Paper 1 can help improve your grade. Now, where's the question? Okay, take your time. Did the question get sent? Oh, okay. Let me. Okay, I'll solve these 2 questions. Here, let's go. This question was annoying. Can you guys see the screen? I think you guys can. Can you guys see the screen? It's me. All right. It doesn't matter. You guys can see it. Someone send the question? Sure. I'll forward the question so you guys can go through it. This was one of the more annoying questions, to be honest. High concentrations of ethanol disrupt the cell surface membrane by denaturing proteins, increasing the separation of adjacent phospholipids. As a result, cell membranes can decrease the thickness by up to 30% and become more permeable. Release ethanol as a waste product of metabolism. In response to increased ethanol concentration, yeast cells are able to increase. Okay, correctly explains a response to ethanol that could account for the increased tolerance. We want to get the ethanol out as fast as possible. Okay, because of this sentence. Prevent the tails on one side of the bilayer from sliding past the tails of the other side of the bilayer. That means that there is less disturbing of the membrane. Since this side doesn't slide past this side, okay, rather than sliding past each other. and causing interference, and later on, perhaps even breaking the entire membrane by the sliding down going up. Okay, we have less sliding, so each remains in its own section, so the membrane remains bound. Okay, and the membrane thickness does not decrease. Again, if they slide past each other, the membrane thickness is going to change and it's going to become more permeable. The problem with B is that while it does increase the fluidity, it also so more unsaturated. 1 second. Here. More unsaturated fatty acids means more kinks. So each tail remains far apart. So each tail remains far apart from each other, so they don't slide past one another. Increase the fluidity, the membrane from being disturbed. In B, okay, we're making the cell surface membrane shorter, which ethanol does, and this is a disadvantage and we don't want it. So B is wrong because we are decreasing the thickness of the cell surface membrane, which ethanol is going to disturb the membrane. C kills the cell, so it's wrong. Cholesterol makes the cell more rigid, so I can't get ethanol out. I can't release ethanol out, so D is wrong. Okay, and in A, I increase the fluidity, so I can still get ethanol out. At the same time, each phospholipid has a much rigid structure. Each phospholipid on their own takes up more past one another. You know, if you have two straight chains, they're going to easily slide. But if you have two zigzags, it's going to take harder, so the membrane isn't disturbed. That's the answer. Sorry I took so long to get to it. I'm really sorry about that. Okay. Yeah, same thing for unsaturated. I'm really sorry about the time I took. Okay. Hello? One second. Okay, I'm going to type the answer. A, cell surface. Okay, for A, we increase fluidity without decreasing length. So for A, we increase fluidity without decreasing length, so less effect of ethanol while still able to get it out of the cell. At the same time, less sliding of the fatty acid chains. Remember, something that has a lot of kinks is going to take a bit more time to slide next to each other than something with than two straight lines. Okay. Less sliding of fatty acid chains, so less disturbance of the membrane and less permeable, less holes in the membrane, less areas where a small molecule can come and disturb it, such as ethanol. For B, first things first, we decrease the length of the membrane, so ethanol has greater effect. We decrease the thickness, of the membrane, so ethanol has greater effect on the thickness. So bad. C is cell death. Bad. D, we are decreasing fluidity, cholesterol, similar action to cholesterol, which decreases fluidity under normal conditions. So yeast cell cannot get ethanol out of the cell. So bad. That's the answer. I'm sorry for taking so long, and I'm sorry for the lag. That's the basic idea. It's a very annoying question. I hope it doesn't come ever again. But yeah. In B, we're decreasing the thickness. So no. Increasing fluidity increases permeability. Sorry, what did I say? Yeah, decreasing fluidity decreases permeability. Yes, this is true. It's okay. I'm going to go through the other questions really quickly. Let's do these. Okay. Which experiment would give a lighter blue 5 minutes compared to the first experiment? All of these except D has the short distance as 0.5. Okay. High temperature, so more of the solution enters. So less. Yeah, at high temperatures there's more diffusion. Okay. And this is compared to the first experiment, which is 1 by 1 by 1. At 10 centimeters cubed at distilled water 20. Okay. So we have either B. The answer is B. Huh. Check this mark scheme and come back to me, because I don't trust. Something's wrong here. Check the mark scheme for 17. 18. Which description is correct? A chromatid is one of two identical parts of a chromosome, is made of proteins and 2 molecules of DNA. Each chromatid has 1 molecule of DNA. I emphasize this multiple times. So B is wrong. Telomere is a sequence of DNA nucleotides. Yes. Repeated many times. Found at the ends of each chromatid. Yes. Ends, notice the plural form of each chromatid. 40. Tetanus is a bacterial infection. Blood antibody concentration, 2 people. On day 0, they were injected with antibodies, and person H was injected with a vaccine. Tetanus antibodies would be produced in tetanus antibodies are not produced in person G. It is passive immunity. He is given the antibodies already. The body does not do anything. For B, antibody production would peak again after if they were infected again on day 20. Tetanus antibodies would not be produced. I misread the question completely. I'm so sorry. If G and H were infected with tetanus on day 20. Okay, so afterwards, G has passive immunity, so they do not build immunity. Okay, however, they will produce antibodies eventually. So A is wrong. For H, we know they took around 12 days to get to produce antibodies. Person H took 12 days to produce antibodies for a primary infection. Okay, so G would take around the same time. After day 32, 20 plus 12 in person G. Completely sensical. H produced around day 13, actually, if we're being more accurate. So after day 32 in person G, because it's going to take the same amount of time. First infection. In C, antibody concentration would stay constant in person H? No. Antibodies are short-term. They will die, but that's why we have memory cells later. Well, memory cells that get converted to plasma cells, that is. Second antibody peak would occur in person H that would be lower than the first peak. No, it would be higher than the first peak, because I already gained immunity, and now I have more memory cells. So when the infection comes again, I am more prepared and I produce more antibodies. So our answer is indeed B. Makes sense. I'll explain it again later. And that's it. Thank you all very much. That's the end of the session. G doesn't have memory cells, so A should be right. It's so if you get an infection, do you just die? If you get a cold, if a baby gets a cold and doesn't have the antibodies against it, do they just die, or do they eventually make antibodies? They'll just like H will eventually make antibodies. It just took time. It just takes time. Okay, in H had no memory cells initially. Okay, and it took him 13 days to make antibodies. G will take around the same time on first infection. Okay, so but H has memory cell. Okay, H will do it faster. Memory cells can be converted to plasma cells. Which explanation? Okay. This one. First things first. Anyone who gets infected will eventually produce antibodies. Eventually will produce antibodies. Okay, it's just going to take time. They're going to undergo all of the humoral response as normal, and they will eventually produce antibodies. H already took the vaccine, and they took 13 days. So the primary infection, they took 13 days to make antibodies. G will have the same situation. They will get a primary infection, and they will take around 13 days to produce antibodies. Okay? But they will produce antibodies eventually, just because H does not initially have memory cells. H, at time 0, they do not have memory cells. They made their own antibodies. Afterwards, they have memory cells. G will be the same. At day 20, they don't have antibodies. They will take their time, but they will produce memory cells. 4B. Okay, antibody production would peak after day 32. Correct. H, under a primary immune response, first time being exposed to the tetanus antigen, took 13 days. G, first time for his body to be exposed to the tetanus antigen, will also take 13 days. B is correct. C, wrong. Antibodies get broken down, but that's why we have memory cells, to make plasma cells, or to be converted to plasma cells, to make us more antibodies. So antibody concentration does not stay constant. A second antibody peak lower than the first peak? No. If I have memory cells, I'm more prepared. I see the tetanus antigen coming from far away. I come with a huge army of multiple plasma cells from the old memory cells that will complete